this video, we're going to have a go at a proton NMR spectroscopy question, and it asks us to determine the number of sets of equivalent protons in the compound below, which is drawn out for us, and their relative signal areas, so how much are they integrated by, which means what's the area under the curve, and their position upfield or downfield. So when we are looking at proton NMR spectroscopy or NMR spectroscopy in general, whether that be 1 hydrogen or carbon-13, we look at three different things. The first thing we look at is the chemical environment, which means when you rotate each hydrogen 1, so one proton, or the carbon-13 atom, does it see the same environment around it? Does it see the same environment? That is, first of all, you have to make sure you can even rotate it, otherwise it might be in a double bond. If it can be rotated, do they see the same things? The second thing we have to look at is integration. Integration is the area under the curve, and it has to do with how many protons in this case, or how many carbon-13 atoms have the same chemical environment. Okay, how many of them have the same chemical environment? Number 3, we look at their upfield or downfield position, which has to do with how shielded it is. How shielded the nucleus is in particular by its electrons, or whether it's not shielded at all. So whether a proton or whether a carbon-13 atom is shielded is dependent on whether it's close to any electronegative elements. So whether it's close to oxygen, nitrogen, or fluorine. If it's close to one of those three, those are very electronegative and they will attract the electrons from its neighboring atoms, which means the hydrogen or the carbon becomes relatively de-shielded. Otherwise, if there's no electronegative atoms that are attracting the hydrogens away from the proton or away from the carbon-13, then it's relatively shielded instead. So the first thing we have to look at is, what are the equivalent protons? In this case, we have to draw in all of the protons in the first place, and it's already drawn in for us. Now, oxygen has 2 lone pairs, so we don't need to draw in anymore for that. And right here, there's a carbon, and there's a carbon here as well, and each of the carbons already have 4 bonds attached to it. So all of the hydrogens are drawn in for us. There's a hydrogen here, a hydrogen here, 3 hydrogens here, and 3 hydrogens here. Now, we have to look for the equivalent protons, which means which ones have the same chemical environment. Well, the first one that has a chemical environment would be this one right here. Now, the only other hydrogen that's on its own is this one right here. However, they do not have the same chemical environment. And the reason for that is because if you look at this first hydrogen, it's bonded to a carbon that's double bonded to an oxygen. On the other hand, this hydrogen right here is bonded to a carbon, which is bonded to 2 methyl groups. So they do not have the same chemical environment. If you were to rotate them, they do not see the same thing. Which means this hydrogen on the left can be considered as a proton with its own chemical environment, so there is no other equivalent proton, and this other hydrogen is another proton with its own chemical environment. And hopefully you realize that the only protons with equivalent chemical environments are the hydrogens that are bonded within these methyl groups. Because those methyl groups are singly bonded to the carbon that's in the middle, this one right here, when you rotate them, they see the exact same thing. Furthermore, those 3 hydrogens itself are each bonded to that carbon in the middle, so with that, it also sees the same thing. So you have 2 hydrogens, or two protons, we should say, two protons with unique chemical environments. Let's just call it CE, with unique chemical environments, and we have 6 protons that are equivalent, which means they have the same chemical environment. Now, integration is a measure of how many protons have the equivalent chemical environment. So in this case, the signal that's caused by this hydrogen here, because there is no other equivalent proton, it will be integrated to 1. Likewise, with this hydrogen on the right-hand side, it will also be integrated to 1 because there is no other equivalent proton. On the other hand, these 3 right here will be integrated to 6. So the last thing we have to figure out now is their positions upfield or downfield. And we can do this 2 different ways. We can do it from our understanding, our theoretical understanding of what it means to be upfield and what it means to be downfield, or we can use a graph that sort of predicts for us already the positions of certain groups of certain sections of the molecule, that whether it would be upfield or downfield. So first of all, let's start off with the theoretical side just so we can implement our theory, okay? Just so we can practice our understanding as well. Now, if something is downfield, it is more de-shielded. If it's de-shielded, it means that the electrons are sort of moving away from the nucleus. So the electron density is spread further apart from the nucleus. And the only reason that would happen is if it was close to electronegative elements. And likewise, if it was more upfield, it would be not close to electronegative elements. So we're talking about your oxygens, your nitrogens, and your fluorines. Okay? If you have a look at the structural formula of this molecule again, what you'll find is that the proton that is closest in proximity to an electronegative element, in this case the only electronegative element we have is oxygen, the only proton that is in close proximity to the oxygen is this proton right here. So you can imagine that this one is relatively downfield. Now, we only have 2 other ones to consider. We have this one and we have these 2, which are having the same chemical environment, so we can consider them together. And if we were to break this molecule up a little bit more, let's zoom in a little bit here. What you'll notice is that one group of protons is a lot further from the oxygens than the other ones. Now, let's show what I mean by that. When you have a methyl group, it's actually the carbon bonded to 3 hydrogens like this. So if we were to expand this out, it would end up being carbon, hydrogen, 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 carbon, hydrogen, hydrogen, hydrogen. So what you'll notice is this. This hydrogen is 1, 2 away from the oxygen, while this hydrogen is 1, 2, 3 away from the oxygen. Which means that all the hydrogens in the methyl groups are a lot further apart, or 1 extra atom apart from the nearest oxygen or electronegative atom, whereas this hydrogen is a tiny bit closer. So you can imagine that when you get the signals for these protons, this hydrogen right here would be relatively more downshield, whereas this one would probably be most upfield. So downfield and upfield. Now that we have this information, let's go and look at our graph to confirm or maybe rebut what we've decided. So in the first case, we have sort of a aldehyde functional group. So there's a carbon that's double bonded to an oxygen and bonded to a hydrogen. So if we go down here, we'll find that it's this group right here, and it is relatively downfield. So downfield is this way, and upfield is this way. So we have confirmed that our conclusion was correct, or our hypothesis was correct, that this would be the most downfield group. Then we said that this hydrogen right here would be more downfield than the hydrogens in the methyl group. And let's see whether that is true. Okay, so what we found is this one right here, which has a signal between 3 to 4 ppms. Whereas this hydrogen right here is not directly bonded to an oxygen, it's bonded to another carbon. Okay, so in that case, we are looking at this signal, the hydrogen bonded to a carbon, which is bonded to another carbon chain. When that happens, we do see, in fact, that this is the most upfield signal. And as a result of that, we have successfully determined whether something is downfield or upfield using our theory, and we validated it using the graph. Now, I hope you all enjoyed this video and you found something helpful in terms of practicing determining chemical environments, how something is integrated, and determining whether something is more upfield or downfield. If you did like this video, please make sure to subscribe to the channel so that you can see more videos on things like this, including chemical engineering as well as chemistry concepts. And please like this video as well, and I'll see you in the next one.